Hi, everyone. My name is William Yu. I am a new assistant professor in the Computational Biology Department at Carnegie Mellon University. I'm very excited to be here, and I thought I'd take a few minutes to share with you some of the exciting work that myself and my lab do. So there are three main sort of themes in the kind of work I do. So I mostly work on algorithms for either genomics or medical informatics. Mostly on the genomics end, but lately I've been moving more into medical informatics. And let me give a couple examples of questions in those realms. So the first one, and the sort of one that's most near and dear to my heart, is rigorously analyzing bioinformatics heuristics. So for example, suppose that you had a genome. Let me do this in black. So let's suppose you had a genome. Let's say it's ACGT, GATTACA, etc. So this would be an example of a DNA sequence that might be found in your cells. And let's say it ends in CCGGTT. So this might have linked, say, 250 million for chromosome 1, or slightly shorter for some of the other chromosomes. Now, one of the standing problems in bioinformatics is how do you quickly figure out how similar one genome is to another? Why might you care? Well, it turns out that measuring similarity between genomes is one way to figure out the sort of evolutionary history of genomes. Because if you recall from biology classes, when a genome mutates, what happens is you might take this G here and turn it into a C. You might take this C here and you might just delete it. You might take this, you might insert something between those two C's there. Let's say you insert an A there, and so on. And so now the question is, after you've done this, you end up with some new sequence, ACGT, and instead of a G, you have a CATTA. You have a blank space there, A, and so on. And at the other end, you have a CACGGTT. Now, one way of determining the similarity between genomes is to say, oh, well, genomes are similar if they don't have very many edits to go from one genome to another. Unfortunately, this turns out to be a very slow operation to compute exactly. It's O of n squared, which is a fancy way of saying that you have to multiply together the length of the two genomes you're aligning together to figure out that they are how many differences there are between them. Now, this is rather slow. So for small genomes, it's feasible, but we have a ton of genomics data now, and this is no longer very straightforward. So instead, people have come up with shortcut methods. One of the shortcuts is something called seed-chain extend. And this is fairly straightforward. Instead of looking at every single position to try to figure out whether or not they're similar, you instead just find substrings that match. So maybe that ACGT matches, that AATA matches, maybe this GGTT matches, and maybe there's some other matches in here. And so that would be the seeding step. And then you combine those together into a chain on both of them. And then you only need to figure out the differences between the anchors. And this turns out empirically to be approximately O of n log n empirically. But how do you actually show that this is the actual runtime? And it turns out that in the worst case, you can't actually show that this is the runtime because you really do need O of n squared time. However, in recent work, my brilliant grad student, Jim Shaw, and myself proved that you can't actually show this in the sort of average case for similar genomes. And this is important for bioinformatics because most of the genomes we care about are the similar ones. That's precisely why we care about looking at them. So this is one example of a problem that I work on. Some of the others that I work on include using these sorts of ideas to do metagenomic comparisons. So metagenomics is comparisons. Metagenomics is the study of genomes of lots of bacteria all in, for example, your gut microbiome. And there are many different ways of doing this. One quick way is instead of just looking at the entire sequences, maybe you just look at collections of these subsequences. So one approach is to take a look at bags of k-mers, which is very fast, but you sort of lose all this linear information. Remember, we have sequences. Another way you can do this is you can look at these chains and look at only the regions that are similar and then compare that. And that turns out to and you have to do this in a sort of clever way because you have to sample the same sets of k-mers on both of the genomes. So there's this coordinated random sampling problem. Coordinated random sampling. But by combining coordinated random sampling with this idea of chaining, you're able to quickly compare together entire swaths of metagenomes. So lots of bacterial genomes. So you can quickly do this kind of sampling. Now, of course, we have, this is all in genomics, and we're sort of talking about the ways that different things are similar in computational biology. Now, how does this apply to medical informatics, which is one of the other areas that my lab studies? Well, it turns out that you can think about different ways of similarity in terms of patient sets as well. So patient set similarity, for example, how many patients have hypertension at 2 different hospitals? So let's say you have hospital A, you have hospital B, and you want to know how many patients have hypertension at both of them, hypertension, or any other medical condition. Now, unfortunately, there's some amount of duplication between hospital A and B potentially because the patient might go to both hospitals, and so you need to somehow deduplicate. Unfortunately, there are privacy concerns with deduplicating because in order to deduplicate, hospital A might have to tell hospital B, hey, I have this list of people who I think have hypertension, do they match yours? And unfortunately, this reveals that those patients have hypertension. And this is a place where coordinated random sampling comes into play. So one way to sort of minimize the amount of privacy loss is to say, well, maybe instead of looking at all my patients and asking you if you have lows, maybe if I just subsample, say, 10,000 of them, and then ask you how many of those 10,000 have hypertension, you can quickly figure out the amount of this intersection, allowing you to compute the union cardinality. And there are a couple different ways of doing this. One common way that we use is we use hash functions to do a coordinated random sampling, even when we don't have direct communication between the 2 hospitals. And so this reduces the sort of random sampling, creates a smaller patient set, reducing the amount of private data that's leaked. And lastly, another thing you can do is once these data sets are small enough, you can apply modern cryptographic techniques in the form of homomorphic encryption, which is a technique that allows you to do computations on encrypted data, but is fairly slow, so it's hard to do on large data sets. So you can apply homomorphic encryption to these smaller subsample data sets to privately compute the similarity between patient sets, allowing you to quickly do federated clinical queries. So in summary, these are a few of the different directions that my lab works on. They're all related to this idea of developing fast or efficient or private algorithms for computing the similarity between 2 objects, whether that's genomes, metagenomes, or patient data sets. I hope this was interesting to you. Thank you all very much.